Welcome back to the second part of Chapter 13. We are going to be continuing our conversation about how cells obtain energy from food. And just a quick recap, by the end of Part 1, you should be familiar with the concept of cellular respiration, also the three steps that are involved, the glycolysis, the citric acid cycle, also known as the Krebs cycle, and the electron transport chain. And then we've also discussed the fact that glycolysis takes place in the cytosol of the cytoplasm. It does not require any oxygen, and its products are going to be the production of ATP molecules, NADH, and 2 pyruvates. So that's where we left off our conversation with the fact that your glucose molecule has been broken apart, has been cleaved, and is now in the form of 2 pyruvate molecules, each with 3 carbons. Now, let's go ahead and see what happens to the pyruvate as we travel down the remainder of the steps for the aerobic respiration. Now, the first thing we're going to see is that our pyruvate has to migrate from the cytoplasm over to the mitochondria. And that is because the two remaining steps of the aerobic respiration, the citric acid cycle as well as the electron transport chain, will both take place within the mitochondria. Now remember, it makes sense to go to the mitochondria because it's the powerhouse of the cell. It will be able to generate ATP molecules. Now, even though both steps take place in the mitochondria, it turns out that they'll use different sections of that organelle. So your citric acid cycle will use the matrix of the mitochondria, where the electron transport chain is going to utilize the inner mitochondrial membrane. And I'll show you a picture of that in just a little bit. Now, because the pyruvate is located in the cytoplasm or the cytosol, we need to go ahead and we need to move it into the mitochondria. And what we see happening is as the pyruvate is pumped into the mitochondrial matrix, something happens with its chemical structure. And you can take a look at it over here. But basically what's going to happen is that the pyruvate will go through what we like to call decarbylation. And decarbylation basically means that we're going to remove one of its carboxyl groups, and along the way we will be able to generate carbon dioxide as a waste product. This process is going to be assisted by the pyruvate dehydrogenase enzyme. And like I said to you before, as the pyruvate moves or transitions from the cytosol into the mitochondria, we see that it's broken down into acetyl-CoA. We're able to generate some carbon dioxide as a waste product, and we do also obtain some high energy electron carriers in the form of NADH. So for us keeping track, we've been able to generate NADH in both glycolysis as well as in this transition step as we're converting pyruvate into acetyl-CoA. I also want to mention something that I had said earlier, which is that please keep in mind that glucose is our perfect or our ideal starting reagent for cellular respiration. However, if glucose is not available, we are able to use our other food sources such as the amino acids from proteins or the glycerol and the fatty acids from lipids. And what we see happening is primarily with fat that their components can also be converted to acetyl-CoA, therefore having to skip the glycolysis step because it's not glucose-based. And since it's able to convert into acetyl-CoA, we see that the products of the fat metabolism will be able to go right back into the citric acid cycle without having to worry about the fact that they did not have glucose at the start of the reaction. Now, the picture below right here showcases the fact that the pyruvate is being broken down into acetyl-CoA, and it's going to be done so by the use of these 3 rather large enzymes that are grouped together, and that group, as I was mentioning to you before, is called the pyruvate dehydrogenase. Remember, anytime you see ASE at the end, it's a really good indicator that you're looking at an enzyme. Now, at the end of this transition step, we now have acetyl-CoA, which has moved into the matrix of the mitochondria and is completely ready to go ahead and start the citric acid cycle. Now, I want to tell to you about 2 molecules that you're going to come across as we discuss the next stage of cellular respiration, the citric acid cycle. So let's go ahead and click over. And on this next slide, it will talk about GTP and FADH2. Now, both of these are going to be examples of activated carriers. In fact, what you can do is come on down to A, and as you can see, here's the chemical structure of GDP. And when you look at it, I hope that it looks a little familiar, right? 3 phosphates. We have our sugar. The only thing that's missing is instead of adenine, we have guanine. So that's right, ATP and GTP, as well as ADP and GDP, are almost like distant cousins from each other. They will work in the same exact way in that they will be able to store our energy on the short term. So they are an activated carrier. And FADH2, which you can see right here on side B, is also going to be a high energy electron carrier, very similar to the NADH and NADH2s that you've come across. So don't be surprised if when you examine the citric acid cycle, you see the appearance of the GTP and the FADH2 molecule. Both of them are activated carriers, and they're once again going to help us as we transition the energy from our glucose molecule. Well, in this case now we've been broken down to acetyl-CoA, from our acetyl-CoA molecule all the way down to the complete breakdown of the carbon cycles, which means that we're able to generate less heat and get more free energy to use for energy unfavorable reactions. So in addition to ATP and NADH, you will also see the appearance of GTP and FADH2. Alrighty, so the citric acid cycle. Now, I am more familiar with the citric acid cycle by calling it the Krebs cycle. So on your exam, I'll be sure to mention it both ways. So whichever one you want to study or name you want to remember, that's fine by me. Both of them are used equally often. In this case, the citric acid cycle, the name is preferred because one of the byproducts that will be generated as we start to pull our acetyl-CoA apart is citric acid. So it makes it a little bit easier to remember the component and the name of the cycle because they basically are a play off each other. The citric acid cycle, as I mentioned to you before, will take place in the matrix of the mitochondria, and its goal is to catalyze the complete oxidation of the remainder of the carbon atoms on the acetyl group in acetyl-CoA. So basically what we want to do is we want to take our acetyl-CoA molecule right here, and through the use of 8 steps, we want to completely remove all the carbon bonds. And as we're doing that, we're obviously going to be utilizing different enzymes to catalyze the pathway and the movement through the 8 stages. And we're also going to be able to generate some ATP molecules, some NADH molecules, some FADH2 molecules, and along the way, we will also go ahead and produce carbon dioxide as a waste product. So go ahead and take a look at the illustration that is on the side of the slide, on the right-hand side of the slide, my apologies. And very similar to when we were looking at the glycolysis steps, there is a color code. Anything that is in red shows you how your acetyl-CoA is taking its turn within the cycle and how the 2 carbons are going to be introduced. And then in blue, we go ahead and we see the carbons in the started oxyactate. So this is the one that will be converted into CO2, the waste product. So let's go ahead and let's take a look at these 8 steps and see what's happening with that oxidation of the carbon atoms. Alright, so as I said to you before, this is occurring within the matrix of the mitochondria. And one thing that I want to point out is, remember, the whole goal is to basically take our acetyl-CoA and catalyze the complete oxidation of the carbon atoms of the acetyl group. And along the way, we're going to generate obviously some ATP molecules, NADH and FADH2, and also generate some carbon dioxide as a waste product. Now, as you look at step 1, I want you to notice that the acetyl group carbons are not oxidized directly. However, what's going to happen is that they're going to be transferred. They're going to be transferred from acetyl-CoA to a little bit of a larger 4-carbon molecule, which is labeled as oxaloacetate. So go ahead and take a look in step 1. You see your acetyl-CoA and you see your oxalic acetate. My apologies, my tongue is twisting. Now, as it does the transfer, what we're going to see happening is that it's going to form what we like to call a tricarbonic acid, which is a 6-carbon, and that is more informally known as citrate or citric acid. And that is one of the reasons why the cycle gets its name of the citric acid cycle. But like I said, I also know it as the Krebs cycle. Once you have the production of your citrate, then the other 7 steps are all about oxidizing it progressively so that we can harness the energy of the oxidation process. And obviously along the way, we're going to be utilizing our different carriers, including our NADH and our FADH2. All right, so please take a look at your chart. Notice how you also have the indication of reversible reactions, and you also have the use of the different enzymes in yellow. So we're going to treat this the same as we did the glycolysis, in that we're going to focus on the names of the enzyme. So you can see for step 1 in the formation or the conversion from acetyl-CoA into the citrate, we're going to use our citrate synthase enzyme. And as you enter into step 2, we see that it's more of an isomerization reaction. So the water that was removed is going to be added back, and then it's going to allow the movement of the hydroxyl group. And in order to accommodate that, we're going to go ahead and take a look at the acetyl-CoA enzyme. Moving into step 3, where we're going to have our isocitrate dehydrogenase. And this is basically going to be part of an oxidation step cycle that we're going to come across. The carbon that you see carrying the hydroxyl group is going to be converted into a carbonyl group. And this is going to be a little of an immediate product, which is very unstable. And this is one of the reasons why we start losing carbon dioxide. This is our little waste product that's being generated, as I said to you before, within the citric acid. And as you continue on to step 4, you also see that by utilizing the ketoglycolic dehydrogenase complex, which is a group of enzymes, we are able to go ahead and start loading up some of our high energy electron carriers. So here's your NADH molecule that you're synthesizing. And yes, you're correct. In step 3, you also had a generation of some NADH molecules. Step 5 is where we're going to go ahead and utilize the enzyme called phenylyl-CoA synthase. And over here, what we're going to see happening is that as the inorganic phosphate displaces this CoA, it's going to go ahead and form a high energy phosphate linkage to sunnate. And this is going to go ahead and utilize the transfer of the phosphate from GDP to GTP. So here's another activated carrier that we're utilizing. And that is then going to lead us into step 6, where we're going to come across another activated carrier. That is going to be the production of FADH2. This is going to be guided by the sunnate dehydrogenase enzyme. And as you can see, the whole goal here is to just keep oxidizing the carbon reactions so that all the energy that's being held together in their bonds is released into the system, and some of them are transferred over to the high energy electron carriers, some of them in the form of GTP and ATP, and obviously some of it is lost in the form of heat, which is obviously not indicated in the picture right here. Step 7 is that utilizing of adding a molecule of water into the system, and we're going to use the enzyme fumarase. And we're almost done, everybody. Hold on there. And this is our last step, step number 8. And what we see happening here is that we're going to use the enzyme malate dehydrogenase. We're going to be able to generate some more NADH molecule, and our end product is going to be our oxaloacetate, which will completely go ahead and be utilized so that we can go ahead and we can finish our ATP extraction, or I should say our energy extraction from our initial molecule, which was the acetyl-CoA which started off the step system. I should also mention to you that since you started off with one glucose molecule and you ended up with two pyruvate molecule, a complete citric acid cycle is actually 2 turns because of the fact that you had 2 acetyl-CoA molecules that were generated with the transition step from the cytoplasm into the matrix of the mitochondria. Now, another thing you want to think about, and I think this is very interesting, is that so far, in the chapter, we've talked about cellular respiration as an example of a catabolic pathway, meaning that we're taking this glucose molecule and we're ripping it apart and we're utilizing the energy that it has bound within it. Now, what we also see happening is that as you're doing your first 2 steps, your glycolysis and your citric acid cycle, we see that along the way you have a lot of intermediates that are being produced. So for instance, go ahead and take a look at your diagram that's on the right hand side, and you can see right here pictured in blue is the glycolysis step. And we can see that as glucose enters into the system, before it makes its way and is broken down into 2 pyruvate molecules, we have a lot of different intermediate products that can be utilized, that are produced, So for instance, you have your glucose 6-phosphate, fructose 6-phosphates, and what happens is within the cell itself is that they can go ahead and they can extract these little intermediate products, and utilizing enzymatic catalytic reactions, they're able to convert it into into organic molecules that the cell will need for other activities. So as you can see, I can take my glucose 6-phosphate and I can use that as a basis or a precursor to help generate nucleotides. So then those can be utilized to do things like the formation of DNA and RNA. Your fructose 6-phosphate can be utilized for the synthesis of amino sugars, glycolipids, glycoproteins. So what the little red boxes are showing you are basically things that can be derived through an anabolic pathway from the precursors or the intermediate products of your glycolysis and citric acid. And because we're using it to synthesize an item, that would then be an example of an anabolic pathway. So it turns out that glycolysis and the citric acid, even though their steps as they're proceeding through their reaction, is a perfect example of a catabolic pathway. We also see that all the items that they are utilizing can be siphoned off and can be used for an anabolic pathway. So your cells have basically figured out a nice way to balance out the destruction and the building of different molecules. This would be another example, a little bit on a larger scale, of linking together an energy favorable and energy unfavorable reaction. All right, we have made it to the last step of cellular respiration. So we started off with glycolysis. We had glucose go in, come out as pyruvate. Then the pyruvate had to transition into the matrix of the mitochondria, and it became reduced or oxidized, to acetyl-CoA. Acetyl-CoA went into the citric acid cycle / the Krebs cycle and has now completely been oxidized. All the carbons have been destroyed, and along the way we have collected NADH and FADH2 molecules, because those are high energy electron carriers. We've also been able to generate some ATP molecules, and now what we're going to do is we're going to take our activated carriers, our NADHs and our FADH2s that we were able to produce in the glycolysis and the Krebs cycle step, and we're going to go ahead and introduce them into the last part, which is going to be called the electron transport chain. Now, your book likes to refer to this step as oxidative phosphorylation, and that is because we're going to be utilizing a different method to produce our ATP molecules. I'm going to refer to it as the electron transport chain because that is the common name of it. I will definitely also explain to you why it's oxidative phosphorylation, but please keep in mind that the third step in my book, in my notes, is going to be called the electron transport chain, which I like to abbreviate as ETC. Now, first things first, this step, the third step, is going to synthesize the majority of our ATP molecules. This one will give us by far more ATP molecules than the other two. On average, if you look at a glucose molecule, your glycolysis will generate 2 ATPs. Your citric acid cycle will generate 2 ATPs. And then your electron transport cycle can generate about between 32 and 34 ATP molecules. It really depends on what cell you're looking at. So by far, you get the most ATP from the electron transport chain. Its location is in the inner mitochondrial membrane. So if you're looking at a little picture that's been inserted, remember the mitochondria is used as an example of the endosymbiotic theory, meaning that it was a free-living organism billions of years ago. It was engulfed by a larger cell, and its proof that this occurred lies in the fact that it has its own DNA and the fact that it has two membranes, an inner membrane that belonged to the original organelle, and then the outer membrane that the host cell put around it. The outer membrane has no function at all. The inner membrane, on the other hand, will have a function. And what we're going to be able to see here is that within the inner membrane, we're going to set up a series of electron carriers that are going to be embedded within. And as we go ahead and pull all the NADHs and FADH molecules that we were generating in the other two steps, we're going to allow them to remove their electrons, and these electrons are going to pass through the chains in a series where they're able to go ahead and exchange their electrons between acceptors and donors. And along the way, the electrons are going to release small bursts of energy. So the electrons are going to start lowering the energy that they carry. Now, the oxygen in aerobic respiration, the oxygen is going to be important in this particular step. Oxygen is going to be our final electron acceptor. So here's the little oxygen molecule coming on in. And because oxygen is our final electron acceptor, we're going to see two things happen. One is we're going to be able to produce the maximum amount of ATP molecules. It turns out that if you use any other gas, like in anaerobic respiration, if you use things like carbon dioxide, you reduce your efficiency to produce ATP molecules. So oxygen is truly the best final electron acceptor. So it's going to increase the amount of ATP molecules that we're able to generate. The second thing is that because you're using oxygen, what we see happening is that you'll be able to generate water as your waste product. Now, as this is occurring and the electrons are being passed from acceptor to donor, we're going to see that the burst of energies that are being released are going to eventually to be used to pump protons, these little H plus molecules you see figured right here. They're going to be used to pump these protons across the membrane. And as they're pumping across the membrane, they're going to create a gradient where you have an area of high versus low. And that is going to be utilized as an energy source to allow for the phosphorylation of ATP molecules. So the fact that we used a carrier as an energy source to generate ATP molecules is why we call this step oxidative phosphorylation. The other two steps, the glycolysis and the Krebs cycle, were just doing their substrate level phosphorylation. They did not need to rely on additional steps from carriers. But in the electron transport chain, we are oxidizing those energy carriers, the FADH and NADH2, and we're doing this through an electron chain. And as the electron is moving, it will create the proton gradient. And I also want to point out that in order to be completely successful in this step, in addition to utilizing water as the final electron acceptor and relying on the proton gradient, we also see that there's an enzyme that is going to be extremely important, and that is going to be the ATP synthase. And this ATP synthase is what we like to call a universal enzyme, in the sense that every single organism that is able to conduct the electron transport chain will have the same enzyme in the same location doing the same exact function. Alrighty, so let me just double check my notes. Let's see, did I mention everything? electron transport chain. I mentioned the location in our mitochondrial membrane. I mentioned the fact that it requires oxygen. It's going to be the final electron acceptor. The end product is going to be a boatload of ATP molecules, as well as the production of H2O as your waste product. And this is the only step where we utilize oxidative phosphorylation to generate our ATP molecules. Beautiful. All right. I think we're doing okay. Now, we've talked several times about catabolic and anabolic reactions. And what we see happening is that we can go ahead and take a look at this beautiful picture that's been posted for us on the right-hand side. And this represents a metabolic map. And in the metabolic map, it's basically trying to showcase you that the building and the destruction of the molecules happens in a very organized and a regulated pattern. And part of what that indicates, and you can see this on the picture, is that notice that you have circles. The circles are going to represent molecules that are basically in the metabolic pathways of being produced. So these are the ones that we know the most about. And then we also see that the circles are connected by lots of little lines. And the lines are going to serve the purpose as representing the enzymatic reactions. And because the lines connect one circle to the next, they're also going to represent the fact that these enzymatic reactions are able to convert one metabolite into the other. So they are really the connection point that keeps everything running nice and smooth. Now, the reason the metabolic map is also important to kind of point out is because your body needs to have a feedback system set up that will, in essence, control when it has glucose breakdown and when it needs to synthesize glucose. Because at the end of the day, that's the molecule that's going to be so important for us to do our aerobic respiration. So your cells have a way to kind of regulate that, and they can do this through a feedback system. They can utilize different hormones, but also what can be done is that they can go ahead and identify a way to store excess food molecules. So this is where things like starch in plants or fat droplets in cells come into play. They provide reservoirs of energy-rich substances that can quickly be utilized to be plugged into our cellular respiration process in case there is no fresh food, quote unquote, coming into the system. Now, I said to you before that the cell is able to regulate when it does glucose breakdown versus glucose synthesis. So at the most simplistic core, if you want to think about it, glucose breakdown is usually done when the body has actively consumed a meal and we're going to be converting the glucose into chemical energy for the cell, or when the body is actively doing an activity, even day to day, every time we need glucose breakdown because that is our main source of the production for chemical energy. Glucose synthesis is when we see that we have a shortage of fresh glucose coming into the body. So how can we go ahead and come up with an alternative source to introduce it into our bloodstream? So on your screen right here, what I wrote for you on the PowerPoint is that if you come across the term gluconeogenesis, genesis means to create, then what we're doing is we are creating glucose. So it's basically going to involve the same exact steps that we see in glycolysis, with the exception of step 1, step 3, and step 10, because those are irreversible. So the other steps it's going to involve, but it's going to do it in reverse, okay? And it's going to be utilizing most of the same enzymes, but once again, because if you go back to your notes and you see the double-headed arrows, we can reverse the steps easily. So that's what we're going to do. Instead of breaking down glucose and producing our pyruvate, we're going to start with the pyruvate and we're going to build it back up into our glucose. In order to do that, we can utilize different enzymes, as I was mentioning to you before. And in fact, what we can do is go ahead and take a look at figure 13-21. And it shows you an example of two enzymes. So here we have our phosphofructokinesis, and we have our fructose 1,6-biphosphatase. These names, could they make them any longer? Either way, what it's telling you is that both of these enzymes work opposites from each other, and it's going to have, through the use of allosteric interaction, it's going to be able to regulate which enzyme is going to be turned on. So what that means is that if I have plenty of glucose, I want to proceed with the phosphofructokinesis enzyme, actively moving through my glycolysis step and breaking down my glucose on its way over to form my ultimate pyruvate molecule. On the other hand, if I see that I need to go ahead and do gluconeogenesis, then what I rely on is my fructose 1,6 biphosphate phase, to reverse the process and now go into the building step and the synthesizing of glucose. Now, as I said to you before, it will be on a feedback system. So if we see that there is plenty of glucose available, then we do shut down the genesis step, and we have to reverse it. If we see that the cell is running out of glucose, then we will stop the glycolysis step, and we will activate the formation of the glucose molecules from the pyruvate case. So let's see what it says. So the feedback regulates. All right, perfect. We also have sources of glucose storage within the cell, and probably the best one for humans is called glycogen. Glycogen is basically a little polymer of glucose molecules, and you can see them right here. Has a little branch point, and it just kind of sticks together, and your body will store it as granules within the liver and the muscle cell. And the way it's going to work is that on a typical day after you have a meal and you have your glucose molecules floating around, if your body doesn't utilize all those glucose molecules right away, it will go ahead and link them together into glycogen. And glycogen comes in to play during our in-between meals phase. So if it's been a while since you've had food, we see that the body starts to crave glucose. It will often go and it will trigger the activation of the glycogen phosphorylase enzyme. And that enzyme will take glycogen and will convert it into glucose 1-phosphate. And then glucose 1-phosphate is further broken down into glucose 6-phosphate. And this can then be utilized in our glycolysis step right over here. So it's taking the place of, I'm going to call it fresh glucose. Now, on the other hand, what we see happening is that we want to make sure that we're not utilizing our glycogen if we actively have enough ATP and we have enough glucose floating around. So on your notes, I did write that you have your glycogen synthase, which is activated by your glucose 6-phosphate, and at the same time, your glycogen phosphorylase, so the one that's breaking down glycogen, that one is inhibited if you have enough glucose 6-phosphate and ATP within the cell. So this is an excellent example of how you can have two enzymes that are relying on a feedback system, meaning that depending on if you have enough product, they become inactive. So in addition to that, we can also use just an old school negative feedback system in the sense that as soon as the body recognizes that you have enough ATP molecules, it will go ahead and inhibit any additional glycogen from being produced. So it will shut down the glycogen phosphorylase. And on the other hand, if it sees that it doesn't have enough ATP molecule and the signal is being sent out, then we start the breakdown step and we see the activation of the glycose synthase, which obviously is activated by the glucose 6-phosphate, which is one of the intermediate products of your glycogen breakdown. Now, we also see that there are a handful of hormones that can help regulate the cycle between synthesis and destruction of your glycogen molecule. Probably the best known are going to be insulin, glucagon, as well as your epinephrines and your adrenaline. So all of these have been known to affect your glucose level and your glycogen level. And then lastly, here are some other molecules that we can use for storage of excess food and energy. In a long term, we see that if you have any excess energy floating around, then glycogen is not needed. Instead, what we're going to do is we're going to store these excess energy foods in the form of fats. And fats basically are little droplets in the adipocytes. So here's a nice picture of adipocytes, which are your fat cells. There are these little circles right here. You can see the fat droplets go into it. And the more reservoir you have, the more it fills up on the inside, expanding the cells. And as a result, the body expands as well. In fact, I remember reading an article that said that most humans, if we were to compare you that have similar body sizes, that have similar heights, as well as age, they tend to have the same number of adipocytes. The difference is that if you're on the larger side, you're housing more fat, which means that the cytoplasmic content of the adipocytes is larger, so the cell is larger. If you're on the smaller side, then the cytoplasmic content is minimum, allowing the cell to become smaller. And if you are on the large side and you decide that you want to start dieting and you start losing weight, well, you don't really get rid of the adipocytes. You literally just shrink them down. They're always going to be there. They could just change the size, which means that they change how much surface area and volume they take up within the body. Now, plants also are known to store excess energy-rich foods. They are not going to store it in the form of fats necessarily, but they are going to go ahead and utilize starch molecules. Plants obviously are autotrophs, meaning that they're able to generate their own food. In this case, photosynthesis will lead to the production of glucose, and then the plant will turn around and use that glucose molecule and plug it into its cellular respiration. So it is a coupled reaction that will continue on. We do see that plants are able to store some fat droplets as well, but like I said, they prefer to do starch granules in these specialized organelles called amyloplasts. And then what we see happening is that the fat as well as the starch will often be stored within the chloroplast. So here at the bottom of your slide, I have your little chloroplast organelle. Well, not your chloroplast, but for the plant, the chloroplast organelle. And you can see at the little black dark circles, those are going to be the fat droplets, and then the larger white ones are going to be your starch granules. So that is where plants are going to be housing their energy-rich foods. We also see that especially in the seeds, they will have a high level of fat, just because the growing plant embryo will utilize it as it develops and it starts to grow. Alrighty, having said that, I think this might be a good place to stop for chapter 13. So we have another one that's down. Hey, look at us go. Please keep in mind that you are more than welcome to just email me with any of your questions, comments, and concerns. And the next chapter we're going to be tackling is also going to talk about energy. It's going to be chapter 14. So we're going to take a deeper look at the mitochondria and learn more about how our cell utilizes energy. All right, everybody, please enjoy the rest of your day, and we will talk again soon.